Introduction. In eukaryotic cells, the mitochondria is vital in cell respiration. Glucose is broken down into carbon dioxide in the presence of oxygen, which produces energy in the form of ATP. There are 3 different steps, the first being glycolysis, which takes place in the cytoplasm, outside of the mitochondria. The second step is called the Krebs cycle, which takes place in the mitochondria. The Krebs cycle creates NAD and FAD, and then the third stage occurs, which takes the energy from NAD and FAD and produces ATP in the electron transport chain. Purpose. The main objective of this experiment is to measure the rate of cellular respiration, which is done by measuring the rate in which oxygen is consumed. You will work with germinating peas, non-germinating peas, and beads to compare their oxygen consumption. You will be using respirometers containing either germinating peas, beads, or non-germinating peas with beads to visually see the oxygen being consumed. You will also be using KOH, which solidifies the carbon dioxide that is produced by the peas and removes the carbon dioxide from the air in each vial. As KOH removes carbon dioxide from the air in each vial, oxygen is also being consumed by the peas at the same time, which then leads to a decrease of the volume of gas in each vial and allows water to enter the pipette. Along with comparing the oxygen consumption within each group, you will also be comparing the effective temperature on the rate of respiration. Materials: germinating peas, KOH, dry peas, beads, 100 milliliter graduated cylinder, cotton balls, 50 milliliter beakers, 6 respirometer vials, 6 stoppers and pipettes, 2 baths, a thermometer, ice, and a dropper. Procedure. First, moisten paper towels to germinate the peas a day before the experiment. Second, fill 2 bins with tap water. Third, add ice to 1 bath to make it 10 degrees Celsius. Fourth, measure 50 milliliters of water into a 100 milliliter graduated cylinder. Fifth, plop 25 germinating peas into the cylinder to measure the volume. Sixth, rip apart a sheet of cotton into 12 cotton balls. Seventh, fill a 50 milliliter beaker with KOH and soak 6 of the cotton balls with it. Eighth, place a soaked cotton ball into the bottom of each vial, and then on top of it place a dry cotton ball. Ninth, while one person finishes the cotton balls, the other should find the volume of the 25 dry peas, which is shown in the background of this clip. 10th, measure how many glass beads it takes to get to the same volume as the 25 germinating peas. Record data for each volume. 11. Make a sling of masking tape attached to each side of the water baths. 12. Place 25 germinating peas into 1 vial. Repeat this step for a second vial. 13. Place the number of dry beads that equal the volume of the 25 germinating peas in the next 2 vials. 14. Place the amount of dry beads that fills the rest of the volume of the 25 germinating peas that the dry peas did not fill in the last 2 vials. 15. After the 6 vials are filled with the respective materials, insert the stopper fitted with the calibrated pipette. 16. Place respirometers so the pipette is resting on the masking tape and let them sit for a 7-minute equilibrium period. 17. After the equilibrium period, immerse all 6 respirometers entirely into the water baths. 18. Allow the respirometers to equilibrate for 3 more minutes, and then record the position of the water in each pipette every 5 minutes and record data. For our statistical analysis, we use 2 chi-square tests of homogeneity. For the first one, we use the data from the 25 degrees Celsius. To find chi-squared in the calculator, for there is too much data to do it by hand, you have to first plug in your observed values into matrix A. To get to matrix A on your TI-84 calculator, click second x the negative 1, and this will get you to all your matrices. Go over and highlight Edit and press Enter for matrix A. Plug in your data by entering in a 6x3 matrix with your observed values, but if they're decimals, make them whole numbers. Always round to the nearest whole number and never use negative values. Then press second x to the negative 1 again to get you back to your matrices. Go over to Edit and click matrix B. Make it again a 6x3 matrix, but do not plug in any data. Leave the numbers all 0. To finally calculate the chi-square, click the Stat button, go over to Tests, and scroll down to option C, and then press Enter. Plug in your observed as matrix A and your expected as matrix B. To do this, the blinking cursor should first be on observed, and to enter matrix A, click second x the negative 1 and choose A, and do the same for the expected but choosing matrix B. Then press calculate and your chi-square will appear with the p-value. Repeat all of these steps for the second chi-square test using the 10 degrees Celsius data, and make sure to clear the data from the 25 degrees Celsius test before calculating. The null hypothesis is that the germinating seeds would not be different than the non-germinating seeds. We accept the null hypothesis for the 25 degrees Celsius data because the chi-square value was too low, which means the differences in the data were due to chance. We reject the null hypothesis for the 10 degrees Celsius data because the chi-square value was too high, which means the differences in the data were not due to chance. The independent variable in the graph is time in minutes, and the dependent variable is the corrected difference. The independent variable in the graph is temperature in degrees Celsius, and the dependent variable is the milliliters of oxygen consumed. Conclusion. In this experiment, the vial with just germinating peas had the greatest consumption of oxygen. This is a result because germinating peas carried out a more rapid process of cellular respiration than the non-germinating peas. The beads carried out no cellular respiration. The non-germinating peas require less energy than the germinating peas, so the dry peas carry out a slower process of cellular respiration. As a result, less oxygen was consumed in the vials with non-germinating peas than the vials with germinating peas. The higher the temperature caused cellular respiration to occur at a higher rate than the lower temperature, and in turn caused a greater consumption of oxygen.